If there hadn't been nanoparticles, there wouldn't have been a vaccine against COVID. So actually, what is it about? Just remember that the COVID vaccine is a messenger ribonucleic acid.
Can we go over in detail what that is?
Yes, so a messenger ribonucleic acid, it's a nucleic acid that has a sequence of nucleotides, and this nucleic acid, when it's inside the cell at the level of the ribosomes, it will produce a protein, amino acids. The nucleosides will be translated into amino acids, and these amino acids will then produce a protein. And this protein is the vaccine protein, is the one that will induce vaccine immunity. So actually, there are three problems with this messenger ribonucleic acid. The first problem is that it's very unstable if you inject messenger ribonucleic acid intramuscularly, like we do with the COVID vaccine. It will be degraded immediately. Because in our body, we have nucleuses, exonucleuses outside the cells, so that's why it needs to be encapsulated to protect it. Second problem is that the messenger ribonucleic acid, even if it wasn't degraded, doesn't enter the cell. Why? You'll understand it easily, because actually messenger ribonucleic acid is a nucleic acid. It is negatively charged because there are phosphate groups, and the cell membrane is also negatively charged. Negative charges repel each other, so the ribonucleic acid does not enter the cells. And on top of that, it's a macromolecule with a molecular weight that's too high to enter, but in the form of nanoparticles. You have this phenomenon of endocytosis, which is the invagination of the cell membrane. And that ultimately allows the particle with its messenger ribonucleic acid content to be delivered inside the cell. But that's not enough. That's not enough, because actually the particle with its messenger ribonucleic acid will end up in endosomes. And there, the translation into protein can't happen in the endosomes. It happens in the cell cytoplasm where you have the ribosomes. So you need to manage to export this molecule, this messenger ribonucleic acid, from the endosomes to the cell cytoplasm so it can be translated into proteins. And that it's a brilliant guy named Cullis who thought about it in order to do that. We simply need nanoparticles that will acquire a positive charge, so they will be positively charged inside the endosomes. To put it simply, to burst the endosomal membrane and allow this messenger ribonucleic acid to enter the cell cytoplasm. And that's what happened. We also modified the messenger RNA, so that's Katalin Karikó, who won the Nobel Prize for that. But Cullis, he discovered these ionizable lipids. And the combination of these two technologies resulted in an absolutely amazing vaccine, since it probably saved millions and millions of patients. There have been more than 1 billion injections. And we can see very, very clearly that not only do we have a vaccine that is more effective than all the vaccines we know, all the other vaccine technologies we've known up to now. But in addition, we know that these vaccines are very, very low in toxicity. Since now we've done more or less over 1 billion injections around the world, and we haven't really seen any serious side effects. So you see, that's another role, I would say, of these nanomedicines, of these nanovectors. It's to deliver a whole series of molecules at the intracellular level that wouldn't be delivered there spontaneously.
Let me come back a bit to this mechanism, because a lot of people have preconceptions and also fears. By the way, among some common misconceptions, many people think that it changes the patient's DNA, that this kind of technique changes the patient's DNA. Is that really the case? And in what way does the cell actually integrate this portion?
So that's not the case at all, unfortunately. Here you obviously need to have at least a minimum of knowledge in molecular biology. Because in fact, the patient's genome is DNA, and you know that DNA, so our genome ultimately will express proteins. How DNA will be transformed into messenger ribonucleic acid, and this messenger ribonucleic acid at the level of the ribosomes will produce the protein that we need. But a messenger ribonucleic acid cannot enter the nucleus for various reasons. But one of the very simple reasons we can understand is that around the nucleus there is a membrane, and this messenger ribonucleic acid cannot, on the one hand, cross this membrane. And on the other hand, even if the messenger ribonucleic acid were at the level of the nucleus, it cannot form the genome because it's not DNA, it's messenger ribonucleic acid. So this ribonucleic acid, it's DNA, it's deoxyribonucleotide messenger ribonucleic acid. So it's not possible for ribonucleic acid to integrate into DNA.
All right then.
So there's no risk. And besides, to be more pragmatic, it's obvious now with the hindsight we have because we vaccinated a considerable number of patients. We haven't seen any genetic or genomic effects at all, etc., etc. Nor have we seen any truly serious side effects.
To go back a bit to these nanomedicines, how does a nanomedicine avoid being attacked by the body, which tends to have defensive reactions to foreign bodies? How do these nanomedicines manage to get past these different filters?
Well, one answer is the second generation vectors that will prevent this opsonization process. Because once again, this opsonization process I was telling you about is a natural defense of the body. When you have an infection, the body will produce opsonins to opsonize viruses, for example. And that's a molecular marker that designates these viruses as foreign particles, which will then be captured by the sick. Macrophages of the reticuloendothelial system, including the Kupffer cells of the liver. And so if we want to avoid it, sometimes we actually take advantage of this effect to target the liver, but when we don't want to target the liver, we try to prevent this immune defense reaction. Quite simply, as I was saying, by coating these nanoparticles with polyethylene glycols, which will create what's called steric repulsion. That is, they will prevent opsonins from binding to the surface of the nanoparticles. And in particular, they prevent the complement system from being activated because complement activation is, I would say, a very important immune response.
All right, so to come back to you a little bit, because we've talked a lot about nanomedicines, but you've also created different startups actually related to these nanomedicines. Could you tell us a bit more about that? What does that involve?
Well, you know, it's true that for researchers, when they make a discovery or when they develop research, the main goal is to publish. By the way, at the Academy of Sciences. Currently, I chair a committee, which is a committee that develops a little bit, or in any case, encourages what we call open science. That is science that is open. So that everyone can have access to publications, so that if you want to read a publication, you don't have to pay. Well, that's what we call open science. Because it's normal for the citizen who pays taxes and therefore pays the researchers' salaries to also be able to see what these researchers produce by reading the publications. That's open science, but you should know that a laboratory like this one, a laboratory that, well, yes, the institute that I led for a long time, it still requires a huge amount of financial resources. So there are times when you say to yourself, I've discovered something that could really be useful, a new nanomedicine that could be useful for treating diseases. At that point, we don't publish, we file a patent because we think it might interest industrial companies. If we file a patent and the industrial companies, they'll be able to develop the drug, which at the university we can't necessarily do. And not at the beginning of my career, but I was already quite far along in my career. There was this authorization given to researchers to create startups, that is, to file patents and then try to develop those patents. So I created a first startup called BioAliens, which was founded in the late 1990s and which went public in 2005. So it was still a success. So why? Because at the research laboratory level, it was a unit at Paris-Saclay. It was Paris-Sud at the time, now it's Paris-Saclay, associated with the Centre National de la Recherche Scientifique. You have to know that here we're discovering. We do all the preclinical research on cell cultures to see if the nanomedicine works at the laboratory animal level, to see, since it was for hepatocarcinoma, if we're going to reach the liver, if we'll be able to deliver the medicine to the liver. If the medicine isn't toxic to heart tissue, since doxorubicin, so all of that, we do it at the animal level. That's fundamental research. But after that, the next phase before putting a medicine on the market is pharmaceutical development. And what is pharmaceutical development? The first thing is to do what's called a regulatory toxicology study, meaning there are rules before putting a medicine on the market, before starting a clinical trial with a medicine. There are rules to show that it's not toxic in animals, and this has to be done under certain conditions that we don't have. At the university, we do toxicological studies, but not in a regulatory, highly controlled way. So that has to be outsourced. It costs money, so you have to raise capital to be able to pay. Then in the lab, you'll prepare 50 mg, 100 mg of nanoparticles, and with that, you can do a lot of tests. With mice, if you inject 10 mg per kilo, a mouse weighs 20 grams. You see, you can treat an incredible number of mice. You have enough to do all your research. But a human weighs 70 kilos, so there you obviously have to produce much larger quantities. That's not research, that's what we call pharmaceutical development. And so there too, we outsource to specialized engineering companies that will do what's called scale up. And in addition, for humans, these nanoparticles, since they are injectable, must be sterile. So we need to find a sterilization method that doesn't destroy the particles. So before moving on to phase 1, clinical trials, the first phase, we already need to find funding to carry out all this pharmaceutical development. Then what we did was we got authorization to conduct a phase 1 clinical trial. So what does that mean in oncology? It means that we will simply first administer it to cancer patients who have obviously given their consent, but to a whole series of patients. Cancer patients with breast cancer, lung cancer, leukemia, etc. We are not going to do any therapeutic treatment yet. We will simply see what dose we can administer. So what do? We will first give a very low dose, and then we will increase the dose. And with doxorubicin, we know that the limiting dose is when we start to see cardiac toxicity. As I was saying, we increase the dose. And then we see that we reach a point where the electrocardiogram starts to show a small abnormality. We go back down to the lower level, and there we've set the dose that we'll use for phase 2. And phase 2, these are patients that we will start to treat, and they will all have the same pathology, that is a hepatocellular carcinoma, a liver tumor, a primary liver tumor. And we will treat these patients. We will do MRIs to see if the tumors disappear. We will study the survival of these patients and so on. This will be done on about 50 patients, mainly in French hospitals, since it is being developed in France. And then we got some very interesting results because we saw a considerable increase in survival time, and in imaging, we saw the tumors disappear. And at the time, the standard treatment we usually used was chemoembolization, and we had better results than with chemoembolization. And then the third step is phase 3. Phase 3 you will conduct what we call a multi-center trial, meaning in several countries, not all the countries in the world, but you will have it in China, because in China, there are many hepatocarcinomas. Chinese hospitals in China will conduct the clinical trial. In the United States, in Japan, in Canada, in France, of course. So it's a trial that will be conducted in several places around the world. It is very, very expensive to develop a new drug. It's almost €1 billion. So because people often criticize the pharmaceutical industry, but you have to understand that these are huge investments. And so now we're going to monitor the patients to see with a larger number of patients. And if everything goes well, so this lasts for a certain number of years, and if everything goes well, actually, then the drug is put on the market.
And between phase 1 and phase 4, how much time passes?
And phase 3. So between phase 1 and phase 3 for a startup. And that's probably the mistake that was made with our startup. Because you have to know that from the moment you go public, that's when the financiers become interested in your company and take the lead. The researcher has nothing more to say. At first, yes, because he's essential to explain the technology, etc. But after that, it's a matter of business. You have to call things as they are. So we need these venture capitalists, what we call venture capitalists, who say, okay, Mr. Couvreur, that's nice, but what are you bringing to the table? An extra patent that was paid for by the Centre National de la Recherche Scientifique. All right, I'm bringing, I don't know, 5 million euros. How much are you putting in? Me, 10,000 euros, because I believe in my thing, so I'm putting all my strength into it. Well, I'm exaggerating, but everything I have, that's what I'm putting in. All right, well, listen, I'll be the one taking over the management of the company, obviously, since I'm putting in 5 million euros. So you see, from that moment on, the profile of the startup changes. We'll need to hire people, especially pharmaceutical development specialists, since I'm not a developer and have never worked in that industry. So you see, the company is going to change its face. We're going to hire people who know the regulatory aspects well and so on. Well, that being said, a startup, even when it has money, even when it's gone public, moves much slower than a big pharma company. A big pharmaceutical company like Sanofi, like Pfizer, like Roche, and so on, they have much greater financial resources and experience with phase 3, which is the most expensive stage in drug development. And so there, a big pharma will be able to shorten the timelines and recruit patients more quickly. Whereas for a startup, it's going to take much longer. This phase 3, our phase 3 took 7 years, if I remember correctly, 6 or 7 years, which is way too long. But in the meantime, you have other technologies developing, you see. So that's the whole story, the difficulty of startups. Being the first to have an idea doesn't guarantee you'll get your drug. In the market, it's often the one who comes second who knows why it didn't work. This brilliant invention can get around that and can probably have a better business plan, etc. So you see, it's still not simple. Just because we have a good scientific idea doesn't necessarily mean it will turn into a medicine.
Okay, very, very interesting. Listen, we're coming to the end of our interview. There's a question I'd like to ask our guests. It's about reading recommendations. For those who would like to learn more, for those who want to go a little further on this topic, do you have any book ideas you would recommend?
Yes, so I don't want to be impertinent, but you know that I had a chair at the Collège de France, and at the Collège de France, it's absolutely fantastic. Because you have to give high-level courses, but they have to be understandable to the general public, since anyone can attend these courses at the Collège de France. And so I held the Liliane Bettencourt Chair of Technological Innovation in 2010. And we have what we call, so of course we give a whole series of courses, but before giving these courses, we do what we call an inaugural lecture, which is a lecture that lasts about an hour. A bit like the interview we had, but with words that are relatively understandable by everyone, simple, without using complicated medical jargon, for example. And so this lecture is. Then we write a small book that is published by Odile Jacob and is available at the Collège de France.
We'll put it in the description for anyone who wants it. In any case, thank you very much for this interview.
I'm the one who should thank you.
Which was extremely interesting.
Thank you very much.